Now we have our translation lecture, and this is going to be the last of our general molecular biology lectures. By the end of this lecture, you should be able to identify the steps involved in translation, list the enzymes and structures necessary for translation, and define numerous terms including protein, amino acid, polypeptide chain, amino or N-terminus, carboxy or C-terminus, codon, genetic code, ribosome, transfer RNA, Wobble Rule, start codon, Shine-Delgarno sequence, reading frame, open reading frame, acceptor site, peptide, and stop codon. So we already know we have DNA. From DNA, that's transcribed into messenger RNA. From messenger RNA, we have translation, which is what generates our proteins. So proteins are the biologic polymers that carry out all of the functions in the cell. And we need different proteins to do different tasks within our cells. So proteins are made from a linear chain of monomers or amino acids, and they fold up into three-dimensional shapes. So a chain of amino acids is a polypeptide chain. So we have our polypeptide chain, or our chain of amino acids, which make up our protein. And our proteins are going to fold up to form a three-dimensional shape. And this folding allows them to carry out their functions. So proteins need to fold properly in order to work. So there are different proteins within a cell. There are structural proteins, which make up numerous subcellular structures such as flagella and microtubules. There are enzymes, and these are the proteins that carry out chemical reactions. Enzymes have to bind to a substrate, and they have an active site where that substrate binds. There are regulatory proteins which control the expression of a gene or the activity of another protein, and transport proteins that carry other molecules across membranes or around the body. So I already said an enzyme has the active site where the substrate needs to first bind in order for that enzyme to work. Amino acids. An amino acid is a monomer or a subunit of a protein. There are 20 different amino acids that are used to make proteins. All amino acids possess a central carbon atom surrounded by an amino group, a central hydrocarbon group, a carboxy group, and a variable group. And this is a schematic showing these features of the amino acids, where you have your central carbon, you have your amino group, you have your carboxyl group, and you have your variable group. Amino acids are joined together to form polypeptide chains, and these polypeptide chains are bonded together. The amino acids are joined by peptide bonds. So a polypeptide chain is a polymeric chain of amino acids. The first amino acid retains a free amino group, and the first amino acid is called the amino or the N-terminus. The last amino acid is left with a free carboxy group, and that's why the last amino acid in a polypeptide chain is termed the C-terminus or carboxy terminus. So you have two amino acids that are peptide bonded together, and you continue binding acids to form a polypeptide chain. That first amino acid is the N-terminal region, and the last at the end is the C-terminal region. So just like in DNA and in RNA, we consider one end 5' and one end 3'. In proteins, we don't say 5' and 3', we say N-terminus and C-terminus. We already said that proteins, our polypeptide chains, are folded. So proteins need to fold properly in order to work. So a complete protein can have more than one polypeptide chain. Proteins usually also have cofactors that are not made up of amino acids. But just keep in mind that although it's an amino acid chain, polypeptide chain, there is a three-dimensional structure to proteins. So proteins have a primary structure, which is the order of the monomers or the sequence, a secondary structure, which is folding or coiling of the original polymer chain through hydrogen bonding, a tertiary structure where you get hydrophilic or water-loving molecules on the surface and hydrophobic or water-hating molecules on the inside. And then you have your quaternary structure where you have several individual polypeptide chains. Here's an oil drop model showing those water-hating molecules in the middle and the water-loving groups on the outside. There are 20 different amino acids that make up protein, and they're represented using abbreviations, either a 3-letter abbreviation or a 1-letter abbreviation. So here are these 3-letter and 1-letter abbreviations. We have alanine, which is ALA or A, arginine, ARG, or the one letter is R, asparagine, ASN, and N, aspartic acid, ASP or D, cysteine, CYS, and C, and so on. So the 20 amino acids have either a 3-letter or a 1-letter abbreviation depending on what textbook you're looking at. So how are proteins made? Each protein is made using the genetic information stored on the chromosome. So the genetic code converts the nucleic acid language, that messenger RNA, into protein language. There are 20 amino acids, yet there's only 4 bases in messenger RNA. We have our G, C, our A, and our U, our uracil instead of thymine. So your bases to make up your protein are read off in groups of 3, and these groups of 3 are called codons. Each codon represents a particular amino acid. There are 64 possible groups of 3. So since there's 64 possible groups of 3, why don't we have 64 amino acids? And the reason is that some amino acids are encoded by more than one codon. Here we have our genetic code. We have the first letter, the second letter, and the third letter, and how these group together is going to depend on what amino acid is made. So for example, if you have U, U, and U, you are going to get a phenylalanine. If you have a U, a G, and a U, you're going to have a cysteine. There are several stop codons, and you should know what the stop codon is. These are UAA, UAG, and UGA. So how do we decode this triplet system? Here's where ribosomes come in. Ribosomes bind to that messenger RNA that was transcribed from the antisense strand of DNA and translate it. So ribosomes have two subunits, the large subunit and the small subunit. After binding, that ribosome is going to move along the messenger RNA and add a new amino acid to that growing polypeptide chain every time it reads that triplet codon. The details of this differ slightly between prokaryotes and eukaryotes, and we're not going to really get into all of the different details because it gets complicated. The translation rule. In general, but there's always exceptions, there is one gene for each protein. We have adapter molecules, such as transfer RNA. Adapter molecules, like transfer RNA, are going to carry the amino acid to the ribosome. The transfer RNA has an anti-codon at one end, and the anti-codon are the three bases that are complementary to the messenger RNA bases. So you have a codon-anti-codon pairing. There's also the Wobble Rule. Each transfer RNA carries a single amino acid. There are 20 amino acids, but like we said, there are 64 codons to be recognized. So some transfer RNAs can read more than one codon. So the rules allow a less rigid base pairing for that codon-anti-codon pair. So the first transfer RNA base can wobble around a little bit since it's not squeezed in between two bases. Then we have charging of the transfer RNA. For every transfer RNA, there's an enzyme that recognizes both the transfer RNA and that corresponding amino acid. We have the enzyme amino-acyl tRNA synthetase. This is the enzyme that's going to attach the amino acid to the transfer RNA. An empty transfer RNA is called an uncharged transfer RNA, whereas a charged transfer RNA is a tRNA that has its amino acid. The transfer RNA has a cloverleaf structure, which is 4 short base-paired stems and 3 loops. The amino acid is bound at the free acceptor stem. The anti-codon is at the opposite end, and the other two are needed for binding to the ribosome and recognizing the adapter enzyme. So here's a depiction of that cloverleaf structure of the transfer RNA. So in protein, we have our reading frames. The reading frame is one of 3 possible ways to read off the bases of the messenger RNA in groups of 3 to give that codon. The reading frame always begins with what's called the start codon. The start codon is almost always AUG. So when the ribosome comes in and sees that AUG on that messenger RNA, that is the reading starts. So AUG is a methionine. So that stretch of RNA beginning at that AUG is called an open reading frame. That open reading frame is what's going to be translated into your protein. There are several open reading frames per messenger RNA molecule. So you really need to find that correct open reading frame so that you make the appropriate protein. So here's an example of a reading frame. We have the one end. Now this is written up as a DNA. we have our 5' end and our 3' end. So ATG, if this were messenger RNA, it'd be AUG because we don't have thymine, right? So ATG is going to be our first triplet codon, or AUG, and that's methionine. Then CCC will be next, and then AAG will be next, and then CTG will be next. And that reading frame is going to continue until you hit a stop codon, such as TAA, TGA, or TAG. Or if it's messenger RNA we're talking about, it's UAA, UGA, and UAG. So getting started, protein synthesis always starts at the methionine, which is the AUG. The initiator transfer RNA is charged and it's tagged with a methionine, a Formyl-methionine or F-methionine. The F-methionine binds to the start codon. The ribosome binding site at the 5' end of the messenger RNA is going to tell what methionine you start with, because you have your messenger RNA. There might be several methionines in there. So that ribosome binding site is going to bind at the start methionine, and this is the Shine-Dalgarno sequence. That start site is the AUG after the Shine-Dalgarno sequence. So right when that Shine-Dalgarno sequence is read, the next AUG immediately following that Shine-Dalgarno is going to start translation. So we have our consensus ribosome binding site sequence. In prokaryotes, that's called the Shine-Dalgarno sequence. In eukaryotes, it's called the COOH sequence. So at the 5' end, we have our ribosome binding site sequence, and that very next AUG or methionine, that A is going to be the +1 site, and that is where translation will begin. So steps in translation, we have initiation, elongation, and termination. In initiation, the messenger RNA binds to the small subunit of the ribosome. The initiator transfer RNA carrying that F-methionine recognizes that AUG start codon. 3 proteins, which are called initiation factors, are going to help arrange all the components of this. The large ribosome subunit then comes in, joins the small ribosomal subunit, and the initiation factors are going to drop away. In elongation, the ribosome has 2 sites for the transfer RNA, an A site or acceptor site and a P site, which is the peptide site. That F-methionine initiator transfer RNA starts at the P site. Then another transfer RNA carrying the next amino acid enters and binds into the A site. That F-methionine is cut loose from the transfer RNA and bonded to amino acid number 2. Now the transfer RNA number 2 carries 2 linked amino acids. There are elongation factors. So the shifting of the transfer RNA on the ribosome is supervised by elongation factors. So that transfer RNA is delivered to the ribosome, binds into that A site by elongation factor T. Elongation factor T is made up of elongation factor TU and elongation factor TS. Another elongation factor G, oversees that everything is moving properly. Our final step is termination. So the end of the message is reached when a stop codon is read, and that's UGA, UAG, or UAA. There is no transfer RNA to read the stop codon, so the reading just stops. Release factors come in, read that stop codon, and chop off the completed polypeptide chain from the transfer RNA. The ribosome then falls off and separates into its subunits. So in summary, you have your small and your large ribosome subunits. You have your transfer RNA with your F-met that is going to come in and bind to that AUG start site. Then you have your next transfer RNA that's going to carry the next amino acid and it's going to bind to the A site. Then the ribosome is going to move down and the next transfer RNA is going to bring in the next amino acid and the next and the next. A polysome is a single messenger RNA that has many ribosomes traveling on it in various stages of translation. So this whole complex of a piece of messenger RNA that with numerous ribosomes and different proteins being made is called the polysome. So please watch the translation movies. These are really, really helpful. So if you guys go into YouTube, www.youtube.com, and you put in translation or transcription or meiosis or mitosis or whatever it is, there might be a movie. Just be careful. Some of the movies are not very good. So I chose one that I thought was very good because there are a lot of movies in there. But for all of you visual learners, these movies are very, very helpful.